Good morning and good afternoon, everyone. My name is Todd Dickinson with BioNano Genomics, and welcome to our 5th webinar in our webinar series. Today we are delighted to welcome our friends at Kansas State University, who will be presenting talks on BioNano Maps to improve insect genome assembly and their work on the Tribolium assembly. Today's seminar will be delivered actually by two folks. Dr. Sue Brown will head off and introduce the topic. Sue Brown is Distinguished Professor at Kansas State University, received her PhD at University of Missouri-Columbia in genetics, and has been studying evolution of gene regulatory networks, and in particular in the red flower beetle, which hopefully most of you can see beautiful pictures of these bugs up on your screen. And then also Jennifer Shelton, bioinformatics core outreach coordinator for KINBRE, which stands for Kansas Idea Network of Biomedical Research Excellence, will follow up and describe the algorithms and the work in even more detail following Sue's introduction. So with that, I'm going to hand it over to Dr. Brown.
Hello, everyone. Welcome to the webinar. And on the front page here, we have a picture of the beetles I study, and just to give you an idea of their size, these are different mutant lines that we have. You can see the black body color in the center, the wild type is in the right, with the castanium meaning chestnut. And these guys are crawling around on cracked wheat. So they're actually quite small. And we do lots of evo-devo studies comparing their development to the developments that we know so well in Drosophila melanogaster. Oops, okay, my screen is not moving. Hang on. There we go. Now it's moving. Okay. So Tribolium is considered the red flower beetle. It is a pest of stored grain, and we're using it as, and we have developed it as a genetic model organism for studies in development, physiology, and toxicology. As a good model organism should be, it's easily cultured. It has a short generation time and large brood size. It has a very small genome size. I've spent a lot of my scientific career making molecular and visible marker genetic maps and lots of genetic tools. We have balancers, we have deficiencies. I started out making genomic libraries, both lambda cDNA libraries and genomic MAC libraries. We've done a lot of mutant analysis, and we were delighted to show in 1999 that this is the first insect outside of Drosophila, in addition to Drosophila, in which RNAi works well, and in fact it works so well that we get a strong, robust RNA response. And so we do a lot of reverse genetic analysis. I'm actually a developmental geneticist, and so we do a lot of RNAi and a lot of double RNAi to do epistasis analysis. We can also do germline transformation, and CRISPR is becoming a very big reverse genetic tool in Tribolium and other insects and practically every system out there. I was involved in leading our efforts to get a 7X Sanger draft genome that was published in Nature in 2008. And this was the first beetle genome to be sequenced. Since then, we've been trying to improve the genome, but when we started, just to give you an idea of what the genome looks like, it's oh, that's wrong. It should be 200 megabases, not 2,000. That's interesting when you're looking at your slides. And I determined that back in 1989 with a COT analysis. But you can also see the karyotypes on the right. There's 9 autosomes, an X and a Y, and it's really hard to tell them all apart. Except for the very long X and the long A grouping, it's really hard to tell any of them apart. But I was able to show with the COT analysis that we had long period interspersion of repetitive DNA, so we knew it was going to be easy to clone genes and to analyze the genome. One of the things that we did have was a molecular map, a genetic recombination map based on molecular markers. And here you can see the 10 linkage groups that we were able to devise. And even though we do have a fair number of markers, you can see that there's very few markers on the X on the left. There's no Y. And since then, we have been able to come up with a Y bin of some of the scaffolds that have been sequenced and assembled. And you can see that there's a lot of variable marker density in here. These molecular markers were used to anchor scaffolds from the genome assembly into chromosome builds. And so when you look at the information that's available for Tribolium castanium version 3 reference genome at NCBI, you can see that we have quite a few contigs. There are about 8,000 contigs, a little over 8,000. And those were scaffolded into 481 scaffolds, plus about almost 2,000 unscaffolded contigs. Of those 481 scaffolds, we were able to put about 90% of the genome onto chromosome builds in about a third of those scaffolds. So you can see about 2/3 of them, 352, were still unmapped in version 3. Since then, we've been using different techniques to try and bring some of those unanchored or unmapped scaffolds into the chromosome build, and that's what we're going to be telling you about today because we've been using the BNG maps to do that. And now I'm going to turn it over to Jennifer, who's going to describe exactly how we've been doing that. Unless there are any questions at this point, I don't think, Todd, we want questions yet, do we?
We can take them at the end.
Yeah, okay. And here's Jennifer.
Hi, so I'm Jennifer Shelton, and I'm just going to be talking today about some algorithms and filters that we developed here using conjunction with the BNG software to improve the Tribolium draft assembly, and we're using BNG physical maps to do that. Okay, so I'm going to be talking about a couple kinds of data formats that may be unfamiliar to some of you, so I'm just going to give you a brief introduction to the main ones. The first is a Bnx text file, and that's just a text file of the molecules that we get off of the machine, so the length of the molecule and the position of its labels. The second is a CMAP, which is short for consensus map, and I'll talk about two main types of CMAP. The first being an in silico CMAP, so we produce those from our genome FASTA files of scaffolds and unscaffolded contigs. The second is a BNG CMAP. That's a CMAP that we assemble from Bnx molecules. So when we align two CMAPs, generally in silico to a BNG CMAP, we call that alignment format an XMAP. So that's analogous to a SAM or BAM alignment format for alignment of sequences, for those of you who are familiar with sequence alignment. Okay, so now that we've gone over the basics kind of novel formats for BNG data, there's 7 major stages to our assembly process, and this is just a quick outline of them. We start off with data that comes from iris chips. So each iris chip has 2 flow cells. It produces TIFF files, which are converted into Bnx files of the molecules. We know that stretch varies from scan to scan for each flow cell. So we split those Bnx's by scan, and then we map those back to our in silico reference sequence. When we do that, we use the alignment to adjust the stretch on the molecules per scan. And we've actually noticed that we can detect a predictable pattern when we have high-quality alignment for the reference in silico CMAP and a good quality signal-to-noise ratio on our scans. So we've begun plotting quality graphs from that stage. So here on the x-axis is a scan for multiple flow cells, and on the y-axis is basis per pixel, which is basically the stretch of a molecule before we've adjusted it. So when I plot these, what I'm looking for is a predictable pattern per flow cell. So I've indicated the first scan of each flow cell with a purple arrow here, and we can see that typically on a good flow cell, I'll have a peak in the BPP followed by a lower plateau, followed by a second lower plateau. So I'm basically checking this just to make sure that everything is looking good. All of our flow cells are high quality, identify any problematic ones, and make sure that we're getting good alignment to our reference genome as we move into the assembly process. So then the next stage in the pipeline is to merge our adjusted Bnx files into a single file. And we take that single file and then we align that again back to the reference genome. This time what we're calculating is noise parameters. We then feed those noise parameters to the assembly pipeline. And we take the user-defined estimated genome size for that organism, so in this case 200 megabases for Tribolium, and we use that to calculate a default p-value threshold. We then figure a strict version of that p-value threshold and a relaxed version of that, and run 3 initial assemblies to try out a range of parameters. We then typically pick the best assembly out of those and run 2 more assemblies testing a strict and a relaxed version of the minimum molecule length with that's performing p-value threshold. So ultimately, we can produce up to 9 assemblies depending on how comfortable we are with the earlier assemblies. In the case of Tribolium, our favorite assembly was the strict p-value threshold assembly. So let's take a look at our Tribolium assembly results. So we started with a genome FASTA of the scaffolds, and that had an N50 of 1.16, so it had made a little bit of an advance from the TCAS 3.0 that we were talking about earlier. And we had 2,240 scaffolds, and they had a cumulative length of 160 megabases, and so that's about 40 megabases shy of the 200 megabase estimated genome size. We created our in silico CMAP from that FASTA file, and you'll notice the number of scaffolds drops when we go to that in silico CMAP. So we only have 223 scaffolds represented in the CMAP. And that's because you only make a CMAP for things that are longer than 20 kb with more than 5 labels. But even though we've lost like 2,000 scaffolds, our cumulative length has only dropped about 8 megabases. So we're still representing the majority of our assembly. Okay, so we use that in silico CMAP to guide our assembly of BNG CMAP, and from that we produce an assembly with a higher N50, so our BNG CMAP N50 is 1.35 megabases, and it's got only 216 contigs and a cumulative length of 200 megabases, which is exactly the estimated genome size for Tribolium. Okay, so now we have a BNG CMAP and an in silico CMAP. So what we begin to do is generate XMaps to evaluate the relationship between these two assemblies. And I'm just going to show you kind of a cartoon diagram of a simple version and a complex version of an XMap alignment to show you how I generally describe these. So I calculate something called breadth of alignment coverage for the in silico CMAP on the top in green here. And breadth of alignment coverage is the non-redundant alignment length. So here it's 1 plus 1.1. I also calculate total alignment length, which is the same in this simple case. So it's again 1 plus 1.1. And I calculate non-redundant alignment or breadth of alignment coverage again for the BNG CMAP as well. So if we imagine a more complex, alignment, we might imagine something where we have 1 megabase region on an in silico CMAP that's aligning to 2 regions on BNG CMAP. So now we have a little bit of redundancy in our alignment, which would make the breadth of alignment coverage for this in silico CMAP 1 megabase, but the total alignment length 1 megabase plus 1 megabase or 2 megabases. And we don't see any change for the BNG CMAP breadth and total alignment coverage. So I check both of these values once I'm done with an assembly because I can understand certain things, or I can infer certain things about the BNG assembly based on the relationship between breadth and total alignment length. So in this case, the difference between the breadth of alignment length and the total alignment length for the in silico CMAP might indicate that we had genomic duplications in the sample molecules that we extracted, or that the molecules were extracted from. We might have had assembly of alternative haplotypes in the BNG CMAP. This might represent a misassembly, creating redundant contigs in the BNG CMAP. And we might have collapsed a repeat in our sequence-based assembly, so our in silico CMAP that is not collapsed in the BNG map. So it gives us kind of a starting position to help evaluate the difference between these two assemblies. So when I ran that assessment on Tribolium, what I found was that there's a 4% redundancy indicated in the in silico CMAP, sort of like the example we saw before. And there's a very low redundancy for this BNG CMAP. And 81% of the in silico CMAP was covered by alignment non-redundantly, and 67% of the BNG CMAP was covered by alignment non-redundantly. So we suspected that 4% redundancy in the in silico CMAP may be due to assembly of haplotypes in the BNG map, which we haven't verified that yet, but we're interested to see how that pans out. So this is data from Tribolium for chromosome linkage group 9 here. And whenever you see these numbers on a scaffold, I've ordered all of our chromosome linkage groups from X to 10 and ordered the scaffolds within those with the first scaffold to the last scaffold. So numbers that are incrementing up around each other, like 131, 133, 134, are typically going to be from the same chromosome linkage group. When we see redundant alignment in the BNG consensus maps, we typically see 2 maps aligning to 1 region in an in silico map, which sounds like it's potentially haplotype in terms of number. If we take a look at the molecules that went into making those redundantly aligning maps, it also looks like it potentially could be haplotype. So here's the first molecule and the second BNG map at the bottom here in blue. And where the molecule pattern is similar in this region, actually almost identical in this region. But then if you look at the region within the box, it appears that we've assembled, or we have molecules that fit two distinct patterns. So we're thinking that potentially this lower coverage region with a distinct pattern is a haplotype. Okay, so another thing that we do with XMaps is use them to super scaffold our scaffold files. So we do that by producing an alignment, an XMap alignment, and we always align with the in silico CMAP as the reference because that's recommended for the BNG aligners. Then we invert that alignment so that we can find instances where BNG CMAP scaffold in silico CMAP. And we work through all of the alignments that we see, filtering them for quality. So one easy way to filter them for quality is to reject anything with a confidence score below, any alignment with a confidence score below a certain value. But another thing that we want to do is find global rather than local alignment. So we also set a threshold for our potential alignment lengths relative to our actual alignment length. And I'm going to show you a quick example of how that would work for this case. So the first alignment, I calculate the potential total alignment length, that's the black arrow. And then I calculate the actual alignment length with the purple arrow. And the difference between them, the purple arrow represents more than 30% of the total potential alignment length. So that alignment would pass and be considered more global than local. The second alignment would again pass. The third alignment would pass because it's a shorter overall alignment, but it still looks more global. It's greater than 30% of its potential length. And the fourth alignment, once we calculated the total potential alignment length, would fail because it's less than 30% of that. And this alignment would pass. Another alignment between the same molecules, or sorry, between the same CMAP would fail because it's less than 30% of its potential length. And the final one would pass. So there we're really just screening for our highest quality global alignment. And once we have those, we check them for the best scaffolding alignment. So in this case, in silico CMAP 2 has two alignments that are high quality and participate in scaffolding. So we would pick the longest with the highest confidence score and exclude the third alignment. So we run stitch for this first iteration. Now we have captured gaps between in silico CMAP 1 and 2 and CMAP 3 and 4. So we represent that in a Class A file, a super scaffold Class A file, and we can actually iterate this process to take advantage of situations where in silico CMAP scaffolds BNG CMAP. So the second time through this process, now our best scaffolding alignments would be the third and fourth alignment, which would join together in one large super scaffold all of the CMAPs. Okay, so another thing that's possible for Stitch to find is a negative gap length. So we see two positive gap lengths here and then a negative gap length that's predicted based on the scaffolding alignment. So when we find negative gap lengths, what we do is replace them with 100 base pair spacers and include them in the FASTA super scaffold file. So we ran Stitch initially with no restriction on negative or positive gap lengths. And what we did was go through and manually evaluate the results of it. And what we found is that the negative gap lengths may be informative for sequence misassembly. So that's a little bit exciting. So our automated version of Stitch iterated 5 times, and we found more positive gaps than negative gaps, 66 positive and 26 negative. But there are some extremely small negative gap lengths indicated in this histogram in blue in this first run of it. So we also have a manually edited version of Tribolium where we use information from the genetic map to include lower quality joins that BNG also supported. as well as breaking some of the extremely small negative gap links. But the really exciting part about this is what the negative gap links seem to indicate. So here we're going to take a look at the smallest negative gap links, and that comes from this alignment where with scaffold 22 and 23 for one chromosome linkage group, and scaffolds 129, 136, and 137 from a different chromosome linkage group that are shown here. And it's this alignment here between the region of 136 and the molecule that also aligns with scaffold 23 that is driving this extremely large negative gap link. But that same region of 136 also aligns to a Bnx map that aligns with members of its own chromosome linkage group. So in our first pass on Tribolium, we rejected that alignment in favor of the alignment that was also high quality and agreed with the genetic map for Tribolium. However, we are going to evaluate this area in 136 and compare it to scaffold 23 at a sequence level to see if it indicates the misassembly. Okay, so our ultimate product from that, once we rejected that alignment and reran stitch, was 2 new super scaffolds that agree with our chromosome linkage group. And you can see those products above as the top 2 alignments. So here are new super scaffolds aligning to BNG maps, and here they are pre-super scaffolded aligning to BNG maps. Okay, so this is actually one of my favorite negative gap links. This one's a really cool case. If we take a look at this very small negative gap link, we can see that we've got 99 to 102 scaffolds that are from one chromosome linkage group, and they're smack in the middle of them is scaffold 81 from a different chromosome linkage group, and then we go back to the correct chromosome linkage group, 103. So that's only half of scaffold 81 that's aligning with the wrong chromosome linkage group. The other half of 81 is aligning with its correct chromosome linkage group, which is kind of exciting. We rejected the scaffold that pulled it in with the incorrect chromosome linkage groups for now, and included it with its known chromosome linkage group, but we're evaluating if we can split the scaffold at a sequence level and essentially our overall assembly, moving it to chromosome linkage group. Okay, so actually every negative gap link less than negative 20,000 bases was flagged independently through our manual evaluation to be checked at the sequence level for misassembly. So that really leads us to believe that those negative gap links might really help drive assembly improvement. All of the shaded negative gap links that you see right here actually represent negative gaps that I manually rejected and reran stitch. So we got rid of the majority of our large, extremely small negative gaps. And in stitch version 1.4.5, which is the most recent version, we actually reject alignment if their gap links are less than negative 20,000, sorry. But we do list these alignments in the data summary because we think that these might be things that need to be evaluated by the researcher. Okay, so the overall result for our super scaffolded Tribolium FASE is that we approximately quadrupled our N50, so we went from 1.16 to 4.46. We only dropped, I think, around, we didn't drop as much in terms of scaffolds, so from 2,240 to 2,150, but we're really working on the biggest pieces of our assembly currently. And the cumulative length increased slightly, so the major benefit that we got was that increase in contiguity and an understanding of how to reorder the scaffolds and what their orientation should be relative to each other. And another fun thing about this is that I don't typically have a genetic map on a project, and in this case I have the Tribolium genetic map, and overall I would see agreement between the Tribolium genetic map and the scaffolds that were suggested by the BNG alignment, which was nice to see that kind of independent verification of the BNG data and its use. And this is just for people, if anyone is interested in trying stitch, they're actually a little bit more complex settings for minimum % of alignment and confidence scores. So I have a first and a second pass of those. I use the first pass to allow high confidence alignment, like 13 or above in this case of Tribolium, with at least a third of the potential alignment lengths realized. But also sometimes you'll have regions that are correct alignment, but they have low confidence scores because they're not very label rich. So if the alignment length is around 90% or more of its potential alignment length, I'll accept lower confidence scores, like a confidence score of 8. And yeah, just again, lower quality alignments for Tribolium were allowed in the final product if the genetic map also supported their order, and complex scaffolds were broken, usually those very small negative gap lengths for our Tribolium work. So here's an example of chromosome linkage group X. So X was really fun. We went from 13 to 2 scaffolds, and we moved one scaffold actually in with chromosome linkage group 3 because it appears that's where it belongs. So that's right here indicated with a purple arrow. Scaffold 2 from chromosome linkage group X fits in perfectly with chromosome linkage group 3. And another thing that we did on chromosome linkage group X was to move into unplaced scaffolds, so we didn't know what chromosome linkage group these scaffolds belonged to, and now we've placed them with X. And another thing here, you can see another example of a potential haplotype that's been assembled by BNG. So if we take a look at the molecule pileups for that, so the molecules here are in yellow and the BNG CMAP is in blue, what I see is an agreement in the pattern on the molecules in the region outside of the purple box. But within the purple box, I have lower coverage and also the pattern shifts between the two putative haplotypes. Okay, so here's another chromosome linkage group just for examples of what we did with Tribolium using these algorithms. So again, at the bottom is the scaffold aligned to the BNG consensus map, and at the top is the chromosome linkage group super scaffold after we've completed the process. Another thing that you can see here, for example, scaffold 127, it's got an inverted alignment, which is why it looks a little bit like it has a pinched waist or an X. A lot of times with BNG alignments, we can also incorrectly oriented scaffolds for a genome. And for chromosome linkage group 9, we went from 21 original scaffolds to 9. For chromosome linkage group 5, we went from 17 scaffolds to 4 scaffolds, and we identified a lot of regions that we think may need to be worked at on a sequence level, which is kind of fun. It'll be interesting to see how that goes. And that just brings me to future directions. So now we're pretty comfortable with our scaffolding pipeline. We're kind of interested in what kind of changes we can make internal to those scaffolds. So, a scaffold from a FASTA file will have a gap region labeled within, and we've just gotten an SD detect pipeline from BioNano from the computational biology division, and Pollock has been really super helpful in showing me how to use it. So now we're thinking what we're going to do is try to target structural variants that are overlapping with gaps in existing scaffolds to see if we can use that to resize those gaps and also potentially address things like potential misassemblies. So that's our future direction, and we're really excited to start playing with that. That is all I have for this presentation. So I just would like to thank Susan Brown, my PI, and Nick Herndon, who helped with quick development, and Mannion Liu, who helped with the manual evaluation, which is about as fun as it sounds. She was awesome on that. And Michelle Coleman, who does an amazing job doing extractions and running it via Iris. And she helps set up high-quality data to start with, especially for non-model species, although she'd hate me for saying that. And Zachary Seifert, who helped with metric summary scripts. And from BioNano Genomics, Ernest Lamb worked with me a bunch to get me up to date on the best practices for the assembly pipeline. And Wei Ping helped me just a total ton with assistance on understanding data formats, understanding how to use IRIS-Q, understanding pretty much everything that I learned in the first year working with that data. And Alex Sheaf works with me just about every week, if not multiple times a week, to help standardize data analysis of this kind of new output, and also to understand how to use the BioNano software and tools. And in terms of script availability, there's a link here to our GitHub account where everything that we develop is available. And PNG scripts that we use, the aligner, the assembler, and now the SP detect software are available from PNG by request, and slides are available online on SlideShare. And then for our supporters, we'd like to thank Cambry, KSU, the I5K Arthropod project, and the Johnson Center for Basic Cancer Research. So if anyone has any questions for Sarah?
Excellent. Thank you, Jennifer. Thank you, Sue. And yes, we are now open to the floor to questions. The way we do questions on these webinars is you type them into your dialog box, and we'll read them off. So the floor is now open. Okay, the first question is, can the software annotate chromosome ends or other structural features?
Is that a question about the structural variant detection software?
It's hard to say. That's the only information we have on the question.
I would broadly say I think that would be a no. It's not really software for annotation. What we're looking for is, so we have a sequence assembler that's using small, very small pieces of sequence, and sometimes dump libraries, which have a problem with being kind of inexact, but you have a broad range in their length, to assemble a genome. So you end up with many pieces parts that, are accurate in regions where you don't have large repeats or skew simple sequence. But with the BNG maps, we have an entirely independent line of evidence for the order and orientation of data within that genome. So pretty much our focus is improving the quality of the assembly. So we're saying here's an independent line of evidence. Where do they agree? Where do they disagree? How can we use that to rearrange, reorder, identify misassemblies, and super scaffold these two genomes? So it's really more about an independent line of evidence to improve the quality of the genome rather than to label a particular area as, a centromeric repeat or.
This is Sue. So if you know that there happens to be the restriction site in the telomere and you have trouble assembling it because it is repetitive, as telomeres are, the BNG map can help. And I think, Todd, you can speak to that and how it helped in Arabidopsis. I think that's also on your website.
Sorry, Sue, what was on our website we were discussing here?
So on the website, under, I think it's in the news brief, there's a little bit about how the BNG maps help assemble into the telomeres in Arabidopsis. Okay.
Yes, I believe you're correct.
I think I've seen that. I can't describe the project, but I've worked on a project where I've seen a repeat that I think was a telomere. But we don't have any. We just manually say, look at that position, look at that pattern. I think that might be a telomere. That's not something that we have automated software to identify currently.
We have some idea of where some of the telomeres are and some of the sequence for telomeres in Tribolium. We have not looked specifically to see whether the BNG maps get us farther into the telomeres.
Okay, great. Well, we have several other questions. I'm going to keep moving on. The next one is, do you happen to remember the average heterozygosity level of the regions that are resolved into two maps?
So in Tribolium Cascadium, we inbred the line 20 generations before we started sequencing, so we were hoping it was mostly homozygous everywhere, but of course it's not always. And so we're thinking that there's just a very little bit of heterozygosity in this line.
Yeah, it's only 4% redundancy for that alignment, so these are not, this doesn't represent the majority of the genome.
Okay. The next one is, I'm curious about the putative haplotypes. Were the analyzed TCAS Tribolium not isogenic? More broadly, can you explain what causes haplotype assembly? Is it SNP variation or indels?
So in this case, it has to be indels unless the SNP is at the restriction site. And in Tribolium, there are just R regions. Even though we've inbred for 20 generations, there are just R regions that we could not homozygose.
Yeah. Okay, good. The next one is, do you have any idea of how the performance of stitch to the new super scaffold process in IrisView? Does one work better?
So I wouldn't say one works better or one works worse. I would say that IrisView software works excellently on supported samples, especially human. A lot of it's tuned to work with human data. And our software is tuned more to work with unsupported sample and typically non-model organisms. So we tried running our Tribolium data through the hybrid pipeline, but again, I would choose different software for different projects. And when we ran it on Tribolium, it was overly conservative. And for example, where X was brought together into the 2 super scaffolds that also agree with the genetic map using our software, X remained in, I think it was over 5 pieces using the hybrid assembler. But if I was working on a project with human data, I would tend to try to use the hybrid map assembler from BNG.
Great, thanks, Jennifer. The next question from Bruce Kingham is, what are the minimum requirements of sequence-based data in order to effectively use mapping data?
And this is Pollock. I can answer this one. So in general, any NGS contig sequence-based data coming in of at least 20 kilobases in length can be in silico digested into a BioNano CMAP. So I would say at least contig should be at least 20 kilobases long to even perform the in silico digestion. Practically speaking, during the actual hybrid scaffolding process, we would like to see contigs of at least 100 kilobases in length. This would allow us, on average, 10 labels per contig, and we would have enough confidence then to perform a proper alignment and resolve any spurious alignments. Of course, if the more complex genome you have, high repetitive regions, the longer sequencing contigs you do have, the better, the more effective your hybrid scaffold will be.
Great. All right, the next question. Are you able to learn anything from the Y chromosome from BioNano data?
We haven't gotten Y yet.
Okay. Okay, good. The next question. Considering the application of BioNano to next-gen sequencing assemblies, what is the recommended minimum scaffold length to use BioNano? I think Pollock just answered this. 100 kb is ideal. 50 kb and above sometimes can. Some of those will be linked into maps, but 100 kb and above is ideal. And the next question is, since assembly scaffolds have ends, how much flexibility do you allow during alignment of in silico assembly map with BioNano Genomics map?
So the BioNano aligner is unaware of whether something is an end-based gap or sequence data. So it just looks to the BioNano aligner like a region that doesn't have a label. So we played around with trying to use contigs for projects and trying to use scaffolds for projects with breaking scaffolds and feeding those back in. And what we found was that we got the best results using scaffolds, including their ends, and we ultimately want to use the structural variant detection to go in and internally break and resize in gaps that are within scaffolds. But also, when I'm working on Tribolium, I don't have any gaps that are existing in the scaffold files that are large enough really to have much of an impact on the BNG alignment. I have worked on projects, especially recently, where I have started noticing very large gaps that are represented in their scaffold files. And I'm really excited to try to develop a pipeline for resizing gaps within using the structural variant detection tools.
I think the end result here is really effective of when you do have a series of ends in your assembly scaffolds, those scaffolds tend to be longer in general for the overall length. And so we can more effectively go ahead and use those to link BioNano Maps and other NGS contigs together.
Can I say one more quick thing here?
Yep.
Okay. So here, these colored boxes that are kind of translucent over these scaffolds represent contigs within the scaffold. So it's just sort of multicolored boxes. And you can't see it, but there's no visible gap in this. When you have a visible gap using this kind of view, it would be like this spot right here, bright green, no labels, and no translucent box over it. This, for example, looks like maybe a translucent box, but it's actually just a light yellow contig. So for Tribolium and for a lot of organisms, I see the gaps in the scaffolds are actually so small that they don't throw off the sequence assembly, or the CMAP assembly. XMAP alignment, there we go.
Okay, great. We still have more questions, so let's do a couple more here. The next one is, have you by any chance validated the gap size inferred by the BioNano Maps versus your reference genome?
Yeah, no, that's what we're doing right now. We're evaluating them at the sequence level, and we're hoping that for the ones that are very small, that we can detect them using our long-distance jump library.
Okay, great. Next one, have you generated any BioNano data for related species of Tribolium and seen how they aligned to the in silico CMAP?
Yeah, so our closest relatives don't seem to be aligning very well to Tribolium, but we are working on.
Okay, great.
Wait, sorry, we were told that a while ago. It looks like Fremani aligned to the Tribolium CMAP and Madden didn't. So yes, one did, one didn't. Right?
That's good. I think the questions are slowing down. Any final questions? Okay, all right, great. Well, thank you. Fantastic presentations, Jennifer and Sue, and for all the great questions. This webinar has been recorded and will be available on our website in a day or two when we get it posted. And I encourage you to keep your eye out for the next webinar series. If you're not a part of the BioNano community, please join and come to our website where you can sign up for it. And we will wrap here. Thank you very much, everybody. Bye-bye.